Hello, everybody. So at Los Alamos, me and Patrick and the other folks, we are interested in finding any biosignature on Europa surface, and I will be showing some of the preliminary stuff that we are doing and trying to standardize our experiments right now. Yeah, as I mentioned, can we detect biosignatures there in Europa? So Patrick will be talking about OrganiCam. It's a prototype time-resolved laser-induced fluorescence instrument that we are trying to develop to detect biological matters on Europa surface. And I will be talking about the extreme radiation and characterization of bacteria in extreme radiation environment. And what would be the possible experiments that we will be doing in coming year. So Patrick, will you please start with OrganiCam? Thank you.
I'm Patrick Gazda. Don't worry about the pronunciation of the name. So at Los Alamos, we're developing some new instruments for detecting biological material on Europa's surface. So kind of the background is that our colleagues at University of Hawaii developed this this instrument called the Biofinder. It's essentially you have a green laser, pulsed laser, and you're expanding light down onto a number of samples here. And then we have a very fast camera that just collects the first about 100 nanoseconds of the light coming back from the sample. And so generally speaking, biological materials in rocks fluoresce with very, very short lifetimes, like in 100 nanosecond range. Polyaromatic hydrocarbons, they have fluorescence of hundreds of nanoseconds. And then generally minerals have luminescence of microseconds to milliseconds. And so if we take a very short snapshot of the light coming back from the sample in the camera, we basically get an image of the fluorescence of just the biological material. And so an example of this is over here where we have Antarctic soil, which is nominally very low in organics, and then you have beach sand from Waikiki, and you take the image in biofluorescence, and the soil has pretty much no signal, but the beach sand is very bright. And so Los Alamos is now taking that idea and developing it into something you could put on the lander on Europa. So you have our camera up on the mast, and we'd be taking pictures like this of fluorescence, and then we're adding this ability to take spectrum as well. And so you could take pictures of the whole area around the lander or where the arm is sampling materials and things like that. And you would get a spectrum of the area as well. And so a lot of what we're doing at the lab right now is developing a radiation hardened system. And so the lab has a lot of experience doing this. For example, we built ChemCam, which is operating on Mars right now, and we're building SuperCam, which is going to go on the Mars 2020 rover. And so we're using pieces of the SuperCam instrument for OrganiCam, and then we're going to make them radiation hardened. We're using a camera system that's being developed in France right now, this CMOS device, which is actually being designed for monitoring the insides of nuclear reactors. And so this is extremely radiation hardened up to gigarad dose. And so we're going to be putting these things together to make our instrument. And so, as Kum said, what we need to do now is start to develop standards that make sense for Europa. Standards that are of our inorganic and organic materials, and then irradiating them to see what the signals we might see. And so kind of this is the general, so as I said before, we're going to have spectral information, which includes Raman information. This is just an example of a Raman spectrum that we took in the lab at Hawaii, where we took a number of different materials like magnesium sulfate, sulfur, a PAH, and beta-alanine, which is amino acid. We mixed it with ice and then cooled that ice down with liquid nitrogen. The nice thing about Raman is that things like the water ice bands and the bands for CH organics are separated from each other, and they're separated from this mineral and organic fingerprint region. And so if there's things like magnesium or some kind of sulfates, they have a very intense peak right here, and so we'd be able to, if there's sulfates there, we'd be able to measure them definitively. Same thing with other types of materials that might be on the surface. Of course, we're also looking for biological signatures, and so this is an example from the literature, but it's showing kind of how much more complicated the spectrum will be. And there's also this, if you kind of step back from the spectrum, there's this very broad feature here, and this is the fluorescence that OrganiCam is actually going to be collecting. So you can think of it as all the light that's coming out from the sample once you energize it with a laser, and then all that light that comes out is summed up in this broad feature. What we want to know is, now with the experiments that we're just starting this year, is adding ionizing radiation, gamma, electrons, neutrons, protons, what changes will we see to these features? Are they going to, we know that stuff's going to break down over time, but we want to know what is the relationship between dose rate, dose flux, and the signals that we see. If we put these bacteria into ice and other things and brines, what are the changes we're going to see? And so Kumkum's going to be talking about sort of the biological side and what's happening to the cells as they're being irradiated.
Thank you, Patrick. So as you have seen, that most of the stuff has been done on E. coli or isolated amino acids and stuff. But I was interested in another bacterium which has been identified in a can of meat which was sterilized by irradiation, but then it got contaminated. And when they retrieved the bacteria, and this was Deinococcus Radiodurans, which is extremely radioresistant, and it has various mechanisms to protect DNA damage and stuff, and it has been of interest lately in the biomedical science because of their mechanism and how we can extrapolate those in our to protect our patients from radiotherapy and stuff. So that's why I have been doing lately, and I'm interested in this part of my research as well. So Deinococcus, it's a radioresistant. It's like 3,000 times more. It can tolerate more, 3,000 times than human being. 5 gray can kill a human, but it can go up to 15,000. So what we have done here, we exposed this Deinococcus and E. coli both in 2 and 20 gray. This is really low, but I want to have some signature at the low dose end, and then I will go at the very high dose end later. I haven't done that experiment, but later this year we will do that. We expose them to a gamma source, and for 46 seconds for 2 gray, and 7 minutes 36 seconds exactly for 20 gray. And what we have seen, the survival rate. So right after exposure, I did the colony forming unit count to find out, and we have seen that E. coli survival rate dropped to 83% when exposed to 2 gray, and 33% to 20 gray, whereas Deinococcus, there is no change. They were basically the same CFU recovered as before the radiation. So then I looked for DNA double-strand break assay. This is kind of like a generalized a picture of what we will see there. Basically the histone that change, and there is a colorimetric assay that we can do, and then take picture and quantify what would be the exact change. And then the next was to isolate the DNA and do the deep sequencing by next-gen DNA sequencing at Llano Genome Facility. So basically, we isolated the genomic DNA. We calibrated whether the isolation was good or bad, and then we did that with Illumina kit. So as you can see here, that in E. coli it was only one chromosome and smaller. Less complicated DNA, whereas the Deinococcus has 2 chromosomes and 2 plasmid DNA, and they are very highly complicated, and they have several mechanisms which actually protect the proteins and the transcriptome later on, even after radiation exposure, and which help them to come back. So this is a bioinformatic platform developed by our genome folks called H. It's Empowering Genomic Analysis. That's the acronym. LANL, everything goes by acronym, and sometimes I forget. The top part is Deinococcus Radiodurans, and the bottom is E. coli, and we looked for SNP, single nucleotide polymorphism, and then indel, which is insertion and deletion, so different types of mutation rate. And as you can see that there are different changes here, and we are still analyzing the data. We are mapping it to the genome, which is already published, and a lot of works actually have been done to identify the genes which are actually changed by 2 and 20 gray and in between E. coli and Deinococcus, so that we can come up with a map later on. But other folks, they have actually worked on Deinococcus, and they have isolated one gene, this DR0865, and they have done a mutant of that. And what they have shown here, that this particular gene was playing a major role in the in offering the resistance, the radio resistant, and also the mineral tolerance, and which will be very important for us, because Europa being so much high in minerals and stuff as you are projecting. And here you can see that with even the high manganese, right? Yeah. So presence, it can it's losing. The mutant is not growing, whereas the wild type is growing even in the high salt. And then this is the gamma exposure, and this is even the 15K. It's still there. It's not like completely dead, the mutant, but the wild type is far more resistant over here. The next one is the UV protection. It's a little lower, but compared to the wild type, but this is certainly responsible for offering the UV resistance to that bacterium, and this is hydrogen peroxide resistance. And so, as you can see, even at the very highest dose, all the wild type were like really protected. So this is kind of telling us the important clue to design our experiment later, with Europa-like condition and mimicking them and stuff. So as I said, that we will expose, our plan is now to have this bacteria colony embedded in ice or in liquid nitrogen or in brine, and playing with different amount of salts and stuff, and then we will do the Raman microscopy and OrganiCam, and we will collect the spectral signature before and after radiation, and that will help us to calibrate our instrument for future development. So Roger Wiens, you might know him. Of course, you know him from ChemCam. He's the PI of this project. Heather, Adam, and Steve, they are our, instrument people who are working on the radiation hardening of the instrument. And the bio team, Cheryl, Kim, Caroline, and Sam, they worked on the genome and isolation of DNA and in my lab. And Dave and Mike are from the radiation protection group. So we have all these sources, and they maintain the sources, and they are the person who helped me to expose this bacteria. In University of Hawaii, Anupam, Tyro, and Shiv are our partners, and they will be working closely with our instrumentation team to develop the OrganiCam. Thank you for your attention, and I will take the question.
All right. Questions?
Hi, Amy Hoffman from JPL. Two quick questions about the bacteria. Are they spore formers?
Yes, they are, if you give them time.
Okay.
Yeah.
And the second one is, what's known about their temperature sensitivity?
Usually these are like 37-degree temperature, like room temperature, to 37 degrees centigrade, like body temperature. Yeah, I didn't mention, but we will be playing around with kind of freezing them and stuff, but they freeze well in the laboratory condition, and they come back, thrive back. But I'm not sure whether they will be propagating under the conditions, So that's another thing. It would be really nice to see if we can find anything which will be developing, but I haven't seen any change when it's frozen even in -20.
And it doesn't, the cell doesn't, no, wow.
Nothing.
Cool.
It just everything. So we usually, when we do like time experiment, like following the time points and stuff, we take it, freeze it, freeze it, and later on we do the experiment to kind of come back to the same situation as it was.
Awesome. Thank you very much. Really cool work.
Thank you.
How much of the biomaterial do you need preserved in order to detect it through fluorescence? Do you need whole cells? Can you have...
We actually right now trying to do it on isolated DNA, but we will be playing around. But what Patrick has already shown, that you can detect DNA, you can detect cells, and Patrick will continue.
Yeah, fluorescence is extremely sensitive to biological material. In fact, they've done experiments where they see down to single cells. I mean, we have not tested that with the instrument yet, but parts per million is definitely a detection limit that we're shooting for.
Hi, I'm Mark Fox-Powell from University of St Andrews in the UK. You may have said this, so apologies if I missed it, but with the OrganiCam, so the idea is to have imaging for the fluorescence over about 100 nanoseconds and Raman spectra. Are those point spectra, or are you making an image cube with the spectral data? Oh, and if they are points, how do you go about deciding where to point your Raman spectrometer?
I should have mentioned that that we'll have, like that image that showed, we're shining the laser down and illuminating a large area. So we will take pictures like our fluorescence images like that. And then when we see bright spots or interesting spots and say, if a context camera says, "Ooh, let's take a picture of that and do fluorescence on it," we can, we're designing it to be able to switch out to put a little lens at the end of our laser to focus it down at a point, and then take a Raman, very sensitive Raman image or spectrum of just that point.
Hi, Tom Nordheim, JPL. Very interesting talk, very interesting work. The only comment I had is you're using a gamma ray source, right? So you might want to take into account that there are protons and heavy ions bombarding things on the surface of Europa. And since they have a different LET spectrum,
yeah, so this is just like an initial experiment that I have done, but we have planned to do use other sources and kind of taking the samples from one source to another and try to see that if there will be like mix and match type of thing. Yeah, we will be playing around with different stuff. Thank you.
So about the fluorescence and Raman spectrometer, fluorescence, you're using a green laser, right? So that means that you would not actually probe anything that is strongly absorbing in the UV. Rather, you go for only molecules that are absorbing in the green. This may actually give some disadvantage to the selectivity. Can you comment on that? Can you detect everything, organic or biomolecules?
We do notice, well, we've done a lot of experiments in the lab using both UV and 532 nanometer laser, and we find that 532, although it doesn't get everything like what you mentioned, we do manage to see a lot of different biological materials. And it just is one of those trade-offs that there aren't that many UV lasers that have space heritage, whereas a 532 that we're developing at Los Alamos and in France, we will have one that has some, space readiness. You know what I mean? So it's just like a trade-off that we had to make.
Just a comment on that. The 532 will not induce a lot of fluorescence, so it'll be better for Raman. But if you want fluorescence, you want a UV wavelength. So you could do both if you had two different excitation sources.
These will be the last two questions, and we need to move on.
The first image you showed was a comparison of Antarctic soils to some beach sand. Could you detect biomarkers in Antarctic soil? Because, there's penguins.
Well, they don't live in the dry valleys. Well, they might. Yeah, I didn't show this, but an experiment that I did way back when I was in grad school, when we're looking at those dry valley soils, we do see very little bit of fluorescence. So that was a nice comparison, but we can definitely see a small amount of fluorescence in those materials, which, if you then do a GCMS on that soil, you say, oh, well, there's a couple ppm of organic material. Yeah, so we can see that.
So you can find life.
Yes, we can find life on Earth.